Hey guys, it's Miss Adelini, and today we're going to be going through the Unit 2 Part 5 notes, which are about enzymes, a specific type of protein, and a little introduction to metabolism. Metabolism is something that we're going to talk about briefly in this unit, our biochemistry unit, but we're also going to be exploring it in both of our cell energy units, the one on cell respiration, which I believe comes first, and then our unit on photosynthesis. We define metabolism as any of the chemical reactions that happen within an organism, and there are two main types. A catabolic pathway is a component of metabolism that involves the release of energy by breaking down more complex molecules into more simple molecules. For example, when digestive enzymes are used to break down food. An example of a digestive enzyme is amylase. It's secreted in our saliva in our mouths, and it helps to break down starches, which are complex polysaccharides. An anabolic pathway is the other type of reaction within metabolism. Instead of breaking things down, here we are consuming energy in order to build more complicated molecules from simpler ones. For example, if we were linking any type of monomer together to build a polymer. With proteins, we would be linking amino acids since they're the monomers of protein molecules. Question number 1 asks you, how are dehydration synthesis and hydrolysis related to catabolism and anabolism? Well, let's look at catabolism first. When we're actually breaking down complex molecules, that sounds to me like hydrolysis, since hydrolysis involves breaking down a molecule by adding water. Anabolism, in contrast, is like dehydration synthesis, since in dehydration synthesis we are building more complex molecules by losing a water molecule, dehydrating it. The image that you guys are given below the anabolic pathway definition shows an example of an anabolic pathway. Usually, an anabolic pathway does not just involve one reaction. It involves a series of steps to go from a really simple starting molecule all the way to a complex product. And usually, each reaction in that pathway is assisted by an enzyme, a type of protein molecule. Without the enzyme, the reaction would either be too slow or it simply would not happen. Okay. Now we're going to go into the types of energy that can be found within an organism or any object. A really important part of living systems is that they're able to transfer energy from one form to another. It's actually considered a characteristic of life to be able to acquire energy-rich molecules and to break them down. Energy is defined as the capacity to do work or the ability to drive other reactions. Kinetic energy is one form of energy. This is the energy of moving objects. Potential energy is going to be energy that results from position. It could be a high up position, and that would represent energy, since if you're high up, you have the potential to fall down to the earth due to the force of gravity. Also, potential energy might be a result of structure, whereas chemical energy is a form of potential energy that has to do with structure, and it's the energy that's stored in the chemical bonds within molecules. For example, if we were talking about the water molecule, there's going to be some energy stored in the covalent bonds between oxygen and hydrogen. In question number 2, it asks us to describe the form of energy found in an apple as it grows on a tree, then falls, and is digested by someone who eats it. So I would probably break this down into steps. The first step, they say it grows on a tree. So in this case, we're building the molecules that are found within the apples, like sugar molecules. And so initially, most of the energy for that apple would be stored in the chemical bonds between the molecules. So there, as the apple is growing, it would have mostly chemical energy, which again is a type of potential energy. Since it's high up in the tree, it also has another form of energy due to its position, because it will fall due to the force of gravity. So it has the potential to fall down. If it's cut off the tree or if it detaches from the tree and starts to fall down, it's got some kinetic energy since it's moving. Once it's on the ground and gets picked up by someone who starts to eat it, the chemical energy within the sugar molecules in the apple starts to get broken down and used by the person who's eating the apple. Okay. Right now, as we talk about energy, we're talking about thermodynamics, which is the study of energy transformations that occur in living organisms and in non-living matter. So we have two laws that scientists have discovered that really govern thermodynamics and energy transformations. The first law of thermodynamics is known as the law of conservation of energy or matter. And what it says is that the energy within the universe is constant. Energy can be transferred and transformed to different types of energy, like with our apple example, going from chemical energy that's held within the apple and potential energy as a result of its high position in the tree to kinetic energy as it falls. This law says that energy can never be created or destroyed. It always stays constant. It just gets transformed into different forms. The second law of thermodynamics is the one that tends to be a little bit difficult for people to understand. So I'm going to give you an example within question number 3 on the side here. The second law of thermodynamics says that every energy transfer is going to increase the entropy, which is another way to say disorder or randomness in the universe. So I'm going to give you an example. If, let's say, I sprayed perfume right next to me and I was in one corner of a room. Over time, the perfume molecules would start to disperse or diffuse across the room. They started out incredibly ordered because they were packed in really tight by that perfume bottle right when I sprayed. And then their disorder or entropy increased as they spread out across the room. In question number 3, we provide another example. We're asking to use the second law of thermodynamics to help explain the diffusion or spreading out of a substance across a biological membrane. So let's say we're talking about your bloodstream, and you have small blood vessels called capillaries that encircle your kidney cells. Now, the role of your kidney cells is to filter out excess water and wastes from your bloodstream and to excrete it. You pee it out. So let's say you have a lot of water in your bloodstream. Let's say you just drank an entire bottle of water and your body needs to get rid of it. Well, there's a lot of water in your bloodstream at that point. And because there's less water within the kidney cell, the water will diffuse or move across the membrane of the kidney cell into the kidney cell. It started out very ordered. There was a lot of water within the bloodstream in a single location right outside the kidney cell, and as it diffused or spread out across the membrane, its entropy increased. So in that case, we're increasing the entropy. Increasing the disorder doesn't mean you're screwing everything up. Instead, in this case, there's a specific reason that the water is moving across the kidney cell. So increasing the entropy isn't necessarily a bad thing. We do have an equation. It's called Gibbs free energy that's used to describe the energy of a system. And we're going to be talking about this more a little bit later. Your homework next class is going to be to watch Mr. Anderson, who's the guy behind Bozeman Biology. He has a really nice video on Gibbs free energy. So I'm going to let him explain most of it to you. But I'll go into the basics now. A change in free energy of your system is what you're measuring, and delta G, which is really what should be in this equation up here. So I'll copy that little triangle, which means a change. So a change in your free energy. And free energy just means the energy that's available to perform work. So the energy that you can actually use to drive other reactions in your body, like maybe to drive the reaction where we put amino acids together to make a protein. The change in free energy is dependent on several things. The total energy in the system. So this includes heat energy. It isn't just the energy that's available to do work. Also the temperature, and we're measuring temperature in degrees Kelvin. And then S, the entropy in the system. Here it says you don't need to be able to do delta G problems, just know that there's an equation. Actually, we found that on the AP test, they did have examples of Gibbs free energy, so we are going to learn how to do those equations in class, just not today. But this is an equation that's used to model the energy in a system. We've got two types of reactions depending on how their free energy changes throughout the course of the reaction as you're going from your starting molecules, your reactants, to your ending molecules, your products. An endergonic reaction is our first type of reaction. This one requires energy, and it's going to absorb energy in order to go from the reactants to the products. In this case, the energy of the products is going to be higher than the energy of the reactants, since energy was absorbed to get to our products. This means there's a positive change in free energy going from our reactants up to our products. In number 4, it asks you to draw graphs to show the process of endergonic versus exergonic reactions with time passing on your x-axis, going from your reactants to your products, and on your y-axis, measuring the change in free energy. What I'm talking about with those graphs is something like this. This is extra image number 1, and it's going to show, oh, you know what, I screwed it up. I put it backwards. So we're going to look at extra image number 2, which is an endergonic reaction. And that's going to be one that absorbs free energy from its surroundings. Usually it's an anabolic reaction because when you're adding molecules together to make a larger molecule, usually that process requires energy. So here we show the progress of the reaction, or time, on the x-axis, and the potential energy of the molecules, or in other words, the free energy, on the y-axis. So they show us going from our reactants to our products, and they show that we have to actually add energy to go from reactants to products. So our delta G, or our change in free energy, is greater than 0. It's positive. For our exergonic reaction, it's the opposite. In an exergonic reaction, we are releasing energy in the process of going from our reactants down to our products. So we have a negative change in free energy. The products have a lower free energy than the reactants. Let's take a look at a graph of that. We'll go back up to our exergonic reaction graph. Usually an exergonic reaction is a catabolic reaction. Typically, breaking apart a molecule will release the energy that's been held in its chemical bonds. So here we're going from reactants with a high energy down to products with a low free energy. So our delta G is going to be below 0, or a negative value. An example of an exergonic reaction, one that releases energy, is cellular respiration. In cellular respiration, you take a glucose molecule, or C6H12O6, an oxygen, and you rearrange those atoms to form new molecules, carbon dioxide, CO2, and water, H2O. The free energy change in this reaction is negative. It's very negative. You've got negative 686 kilocalories. And that free energy that gets released is available to do other work in the cell. We use the energy from the conversion of glucose and oxygen to CO2 and water to create ATP molecules, an energy-rich molecule that can be used to fuel other reactions. Spontaneous reactions are a type of exergonic reaction that can occur without any energy input at all. So they'll happen spontaneously, randomly, instantly, and the energy that's released as a result of a spontaneous reaction can be harnessed or used to do work. A lot of times the cell will take the energy that's released as a result of an exergonic or spontaneous reaction and use that free energy that's released to power an endergonic reaction, a reaction that requires energy to go from the reactants to the products. ATP, an energy-rich molecule in the cell, is often used for these energy coupling relationships. ATP, its full name is adenosine triphosphate. Adenosine because its basic structure is that of an adenine nitrogenous base, and that's right here. And then we've also got the 5-carbon sugar, which is also seen in a nucleotide. But then we've got 3 phosphate groups instead of 1. So overall, it looks like a nucleotide with a nitrogen base, a 5-carbon sugar, and a phosphate group, but there's 2 other phosphate groups, making it triphosphate, 3 phosphates. This is the primary source of energy in all living things. It's a really good energy molecule. And in order to make ATP, we take the energy that's released as a result of cellular respiration, converting glucose into carbon dioxide and water, and we use that energy that's released to add a phosphate group to ADP. ADP is adenosine diphosphate. It looks the same as this, except hey, it's only got di or 2 phosphates. Adding that phosphate group is going to store energy within this molecule, and if we cut off that last phosphate group, it's going to release a lot of energy. So this diagram shows us how we have to put some energy in order to convert ADP, an inorganic phosphate, to ATP, and how breaking ATP down is going to release energy. Okay. ATP and the breakdown of ATP is an exergonic reaction where we're removing the phosphate group. We can use the energy that's released as a result of breaking off that last phosphate group to fuel endergonic reactions within the cell. Now, there are 3 types of things that we might want to power using the breakdown or hydrolysis of ATP. One type of work would be mechanical work, so the beating of cilia, which are almost like tails found on cells that can help them to move. You have ciliated cells in your respiratory tract, so they have those little waving cilia, and the goal of those cilia is to get foreign particles like dust out of your respiratory tract. Without ATP being broken down, you wouldn't have the energy to fuel that endergonic reaction of beating cilia. We also use the breakdown of ATP to fuel the contraction of muscle cells, the movement of chromosomes, a bunch of other mechanical things within the cell. We use the breakdown of ATP to power transport reactions like pumping substances across membranes against the direction that they would normally move. So when we talked about a lot of water in the bloodstream moving into a kidney cell, that's natural. It's moving from an area where it's highly concentrated in the bloodstream to an area of lower concentration within the kidney cell. But sometimes cells will move substances from an area where they're they have a very low concentration to an area where they have a very high concentration, and that does require some energy. The other type of work would be chemical work, and that would be an anabolic reaction where we'd make polymers from monomers. Okay, so this is just another picture of the structure of ATP showing the adenine nitrogenous base, the ribose sugar, and then our 3 phosphate groups. Okay, and this shows us how ATP can be broken down. All you have to do is add water. And when we add water and break this inorganic phosphate group off, that process is an example of hydrolysis. Add water to break something apart. So this is the hydrolysis of ATP, and in the process, energy is released. An example of ATP hydrolysis being coupled to other reactions involves the creation of the amino acid glutamine. Glutamine is made from two building blocks, glutamic acid and ammonia, and it's a process that requires an input of free energy. It requires an input of 3.4 kilocalories. Now, it wouldn't be able to get that energy if it did not have the energy from the breakdown of ATP. The energy from the breakdown of ATP provides for you 7.3 kcals of energy. So we're able to use that energy in order to fuel the creation of glutamine. And here, I think they may have screwed this up a little bit. Since free energy is being lost, it's being released, this really should be negative 7.3 kcals, meaning it's being released. So when you add those two together overall, we still have some free energy left over to use in another reaction. The synthesis of glutamine did not use up all the energy that was created by breaking down ATP. A lot of times, the way that the energy from ATP is used is by transferring the phosphate group that has a lot of energy associated with it to another molecule. When we put that high energy phosphate group on another molecule, we say that that molecule is phosphorylated and is now more reactive and willing to react with other molecules. Like, for example, if I was to phosphorylate, add this phosphate group to glutamic acid, it's going to be more willing to meet up with ammonia. And they actually show that in this picture right here. Okay, so they show us how with ATP, when we break ATP down, the single phosphate group is transferred to glutamic acid, which makes the glutamic acid want to interact with the ammonia, and then we're able to form glutamine, the amino acid. And at that point, the little phosphate group has lost its energy and it pops off. So the energy that's required to put glutamic acid and ammonia together is 3.4 kcals. And we get exactly negative 7.3 kcals. So the process of ATP breakdown releases 7.3 kcals, and overall we have some energy left over if we want to use it for another reaction. An interesting fun fact is that, within a muscle cell, we make ATP really, really quickly. Over 10 million ATP molecules are used and broken down into ADP and P, and then they're created again by adding that phosphate group back onto the ADP molecule per second per cell. So 100 million ATPs per second per cell are used and then regenerated. Crazy, crazy town. Okay, so that concludes our first segment of the Unit 2 Part 5 notes on enzymes and intro to metabolism. When we come back, we'll talk about enzymes, which are a specific type of protein molecule, and how they help assist with reactions by assisting with energy transformations.